familiar with other kinds of drug products, and cell therapy is an example of a drug product. For example, small molecule drugs are exemplified by something like aspirin, and those drugs are made by chemical synthesis methods. But there are other drugs we call biologics, and that could be, for example, a small protein such as human growth hormone, or a larger protein such as an antibody, or a more complex structure that even contains DNA and RNA like a virus. All of those are biologics, and that means that we use biological processes to produce the drugs. But none of these are quite as complex as a cell therapy, because in a cell therapy, the cells are the drugs. You can see that they differ in several obvious ways visually, and that's in terms of their increasing complexity and increasing size. A small molecule drug can be dozens of atoms, whereas a protein can be thousands of atoms, and a cell can be more than 1 billion atoms. Not only that, the cell is more complex structurally with compartments and different functions and the ability to replicate itself. So with that complexity comes powerful benefits for new classes of medicine, but it also comes with important manufacturing considerations. So in the context of this course, what we mean by a cell therapy is when living, intact cells are produced and delivered as the therapeutic product. The cells are the drug product, and the intention is to use those cells and the functions that they promote to treat or cure the disease of the patient. Let's take a look at a couple of examples. Today, we use immune cells to produce drugs that are intended to treat cancer. For example, you may have heard of CAR T cells as one class of immune cells that's been genetically engineered to use for the treatment of blood cancers. But there are other kinds of cell therapies that are in development all over the world, including using stem cells to treat tissue diseases, and you might think of this as regenerative medicine, or in certain contexts, you might even think that those cells are being used to produce a tissue-engineered product, where the cells are produced in a carrier, such as a matrix, and then those cells within that carrier or matrix are delivered to the patient to produce the therapeutic outcome. Now, in all of those contexts, in order to produce the cells as medicine, what we have to realize is that we're making the cells proliferate, increase in cell number, so that we can get up to the appropriate therapeutic dosage for the patient. So we grow the cells in special environments, sometimes called bioreactors, to increase the cell number while maintaining or inducing the properties that we want the cells to have to be good medicine. And then we take those cells, formulate and package them at the proper dosage, and deliver those cells as medicine to the patient. However, it's important to realize that because cell therapies are so new as a class of medicines, that there are various terms and various definitions that are being circulated. And so as you read through the literature, as you talk within your communities about cell therapy manufacturing, it's important to be aware of those alternative terms or alternative concepts of what we mean by cell therapy. So let's look at an example of CAR T cell therapies. In this course, we would call that a cell therapy because the cells are delivered to the patient as the medicine. But in fact, to produce that therapy, the cells were genetically engineered so that they expressed a very specific receptor on the outer surface, a chimeric antigen receptor, or a CAR. So when this paper came out in 2018 in Nature Gene Therapy to describe this innovation and this newly approved medicine, it was referred to as a gene therapy. Again, because the gene manipulation actually produced the therapeutic benefit, but the cells were the product that was delivered to the patient. In fact, the FDA's own press release referred to these CAR T cells as a cell-based gene therapy. Again, because the cells were delivered, but they were emphasizing this distinction of the genetic modification of the cells. And then when we look at the final approved product packaging, this drug product is referred to as a genetically modified autologous T-cell immunotherapy. All of those terms are correct, but in the context of this course, it is a cell therapy, and those other descriptors are details of the manufacturing process and the cell source, and then the intended target application. Now, another term that you might hear to cover cell therapies is advanced therapy medicinal products, or ATMPs. And this term was coined by the European medicines agencies, intended to cover a much broader range of products, including cell therapy, but not limited to it. So this term, ATMPs, covers gene therapies, cell therapies, and tissue-engineered constructs. So when genes are delivered to the patient, cells are delivered to the patient, or cells within constructs are delivered to the patient. So far, we've covered what a cell therapy is in the context of this course, as well as some other terms you might hear as you discover, learn, read, and start to work in the context of cell therapy and manufacturing. In the next section, we'll cover some additional terms that describe types of cell therapy and a few topics that we won't cover in this course.